I know exactly who is attending and who is not. But Arunakshu, are you there?
Sir, I am here.
Okay, so I should sort of now go to the what is it? Meeting options and allow you to present. So everyone, and I will. So you can straight away take over and show them the slides. So what I was doing was just talking. So is it all right? Can you start? Straight away you can show the first set, which is.
Yeah, I will start now.
Yeah, and you can give them the links, or they will be able to see the links so they can watch it again over the video. And I would suggest that whatever questions you have, you say it, and later on in the same thing, when you close, you can type in these questions if you can, or if you can write it out, whatever you want to do it, whichever, as a small file, upload it. All the questions that you have, that is the guideline of how they have to submit the practical notebook. Is that all right with all of you?
Yes, sir. Yes.
Okay, so Arunakshu, if you have those things, can you just sort of.
So I am starting now.
Yeah, so I should sort of minimize or whatever, you know. Yeah, I mean.
So everybody can see my screen, how Enzyme works.
I think it will make it larger.
Yes, sir.
But you have to make it larger, Arunakshu.
Yeah, I will. So let's start with this video. So this is an introductory one. So everybody please now listen.
Sir, we cannot hear.
Yeah, actually, Arunakshu, you should also give them links. This is.
Mute myself so that they can hear.
Arunakshu, if you mute yourself, I don't know, but we can't hear the video. Can you just increase the volume?
Sir, I am already muted.
You are muted, but I don't know why the video, when it plays inside this, is not. It's not possible to hear it. Do you just want to share the link and everybody watches the video on their own screen? Is that possible with you people?
Yes, sir.
Yeah,
Yes,
so
sir,
just paste it. And he will run it, and you, I don't know, how will you then sort of, how will we run this together? So just paste it as a.
So while sharing, there's an option of share device audio as well. So I guess that isn't turned on.
Okay, Arunakshu, is it possible that is the problem? Just share. Just, you know.
My shared device audio is actually
on, but somehow it's not working.
Sir, on Teams web, there are several options that are unavailable. So if you have the app, then you can start the app.
So anybody has who has the app, you take on and share it. Okay, I will be, and after that I will take over.
Yeah, anybody who has the app, can you just open this video, and because everybody is allowed to share. All of you, any one of you can share the screen. So Arunakshu is there as a facilitator. Any one of you who is taking it over? Vishant? Vishatan?
So it's Vishantan. Yes, I can share. So just a second.
Vishant. It is Vishant. Why is it
So,
Vishatan?
So it's Vishantan, sir.
Vishantan. Okay. That
Yes,
is a
sir.
very, yeah, emphasis is on the first syllable, Vishantan.
Yes, sir.
Okay, thanks. Sorry for this,
So I hope
mispronouncing
it's
your name.
visible.
Yeah. Now, can we hear you? That is the question.
So is it audible?
Every
Yes,
single
audible.
second
Audible.
inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur. To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell, in every living creature on the planet.
Please continue the video.
So I
Please
guess.
let the video. Yeah, वो अपने आप ही चलने दो, अपने आप ही next वाला आ जाएगा. Just run it.
Okay. So just a second. So that's it. So should I start with this one, sir?
Let me interrupt. In your suggestion panel on the
Pathways, such as the citric.
Yes, sir.
so in your suggestion panel, you may be seeing a video by a lady. It is 5 enzyme and catalysis. Can you see on the right panel?
Not really, sir.
MIT open courseware, enzymes and catalysis.
So probably I'll just like search for it. Could you
Just
tell
write
the
it
name
on the
again?
search, enzymes and catalysis. MIT open courseware.
So this one.
Yeah.
Yes.
The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
Okay, so welcome to class today. Today I'm going to be talking about one of my favorite topics, enzymes and catalysis.
So
and what
everybody now stop the video.
an outline of.
Okay, so
Sorry, sir.
now you guys please share the link between you, the previous video and this one, right? So this is a 1 hour video which you have to watch and watch it very carefully. And then I have framed 5 questions which have been discussed in this video. So you have to answer those questions after watching these videos. And you have to answer these questions after watching this video. And I'm just going to discuss 1 or 2 things which are mentioned in this video so that you can answer the questions easily. So now I hope you have shared the link between you.
Yes, sir.
Okay, so now I'm showing you the questions, so please write it down or take a screenshot of the questions. So now Vishant, please stop sharing. I will share now.
Fine, sir.
And also the fact is that if you think the questions are tougher, you say it right here. Okay, if you feel they are, please don't tell if you don't feel. So everybody can see the questions now.
Yes, sir, we can see.
So these are the 5 questions. Just read and tell me if you can answer this. I am sure you will be able to answer these questions after if you have watched the videos carefully. But still, just read it through and let me know whether you have any questions. Sir, are you online?
So could you share the questions again?
Sorry, just a second. Yeah. Everybody can see the questions now?
Yes, sir.
So any doubts regarding understanding the questions?
Sir, in question 1, like the second part, explain with an energy profile diagram. Sir, like in the first video, we saw the diagram of enzyme activation energy. So can we use that diagram to answer that question? This question.
It is a very straightforward question, and you will able to answer if you are watching the video closely.
Okay, sir.
Question number 2.
So the questions are clear.
Sorry, once more.
The questions are clear.
Okay. So question, there is no doubt in question. Okay, fine. So question number 3 now. Just let me know whether if you have any doubt regarding what I am asking. That's it. The answer you have to devise on your own. Okay, so since nobody has any
Sir, sir, the fourth question, sir, could you explain what the question is asking?
The fourth question is asking what is the connection between cell and enzyme specificity or promiscuity. What I am trying to ask is that whether the enzyme is actually specific inside the cell or it is specific in any part of the universe. Like if you take out the enzyme from the cell and put it in a in your reaction mix in your test tube, will it have the same specificity, or its specificity is will be reduced, or the promiscuity is actually the antonym of specificity? So whether the cell is specific inside the cell only, or it is specific outside the cells, or it becomes a little promiscuous when it is outside the cell, and if it does so, why these things happen? Clear?
Yes, sir, clear.
Okay.
Sir, you said promiscuity is antonym of specificity.
Yeah, at least in enzyme science, not
So
every.
means promiscuity means the enzyme is not specific.
Yeah, exactly. Or it is less specific, or it's like it's barely have any specificity like this. Okay. So all these questions have been covered in that lecture by the lady in the MIT Open Courseware. So you have to curate very carefully to answer these questions. Okay, that is point number 1. Point number 2, none of the answers will require more than 50 to 60 words, or at most 75 words. Got it?
Yes, sir.
Okay, so your words should be limited. Yes, the energy you can draw as much as, not as much as, like you can draw maximum 5 figures or 5 reaction schemes, or chalo 6-7 reaction schemes. So your schemes or drawings can be there, but you cannot go more than 50 to 75 words. That's your limit, right? And it should not go above 100, then there will be negative marking. Got it?
Okay. Yes, sir. Yes.
Okay. So any of these things you have never heard of, or if you have any doubt regarding the energy profile diagram, or any fundamental ideas which are required to answer these questions, if you are lacking those, I can briefly explain, or I can clear your doubts. You can ask.
Could you please explain what is promiscuity?
You yourself told now what is promiscuity. It is the antonym of specificity.
Oh, sir, I do not know.
If you are not happy with the word promiscuity, then take the question remove the promiscuity. Still, the question is as much as valid.
Okay, sir. Okay.
Anybody else feel the problem with the word promiscuity? You can Google right away and see. And the lady has actually used the word promiscuity in the lecture video. Okay.
The following.
So again, a little mistake from my part. I called her a lady. That was very bad. We should call her Dr. Stobe.
So one more thing, when and how do we have to submit these answers?
As usual, you have to submit to Professor KJM over the Moodle, and like you have all submitted the experimental things, protein purification and all those things.
Right, so it's basically like the lab report for this week, right?
Yeah.
Fine, sir.
Okay. And please, actually, I saw KJM checking your copies, and he also showed some part of those things. Actually, we told you to write precisely. So most of you have copied, which is not very good gesture, first of all. Second thing is that how many people don't know the disinfectant is a hypochlorite solution or a bleaching powder, not an hydrochloric acid solution. You people don't know this. Got my point? So many people have written that we use hydrochloride to the media cultures for disinfection. That was a big mistake. Another part was this one, your use of mercaptoethanol or DTT. So all of you have written the disulfide bond should be protected from oxidation. The disulfide bond is actually oxidized. We are using the mercaptoethanol or DTT to reduce the disulfide bond while we are purifying, so that they don't reshuffle and make wrong disulfide bonds. If you are telling that it is protecting the protein from outside oxygen attacks, that's fine. But you are saying that we are protecting the disulfide bond while the disulfide bond is actually oxidized. So what is the reduced form of a disulfide bond? Anybody? What is it for?
Individual cysteines.
I am asking, when you reduce the disulfide bond, what you get? What is the name of that group you get? Anybody?
So thiol.
Yes. Perfect. That's it. So when you oxidize a thiol, it becomes a disulfide bond. So you are not protecting the disulfide bond from oxidation using your mercaptoethanol or DTT. Got my point? So these are the mistakes you should not repeat, because like, obviously now you are on a stage where this type of mistakes cannot be allowed, and many of you people have copied. It is very clear. Same line. Even I am telling you, in some question papers, answer sheets, the pH, the P is small, the P is small, the H is large. Everybody knows that, but people have written pH, small p, small h, and the same pH mistake is there in multiple copies. How that can happen? That cannot be a random mistake, no? There's a specific pattern. I hope you are taking note of my points. Hello, anybody there?
Yes, sir.
Yes,
We
sir.
are, yes, sir.
So I don't, but please don't do this, because after this KJM is going to penalize you more. That I am sure.
Sir, we have to submit this report and the previous two reports together this week on 20th.
Now that you ask Professor Mukherjee directly, because here my part was to show you the video, and I have sent you call him and ask him how you should submit. And I hope I made myself clear for the points I have seen to you. And we have also mentioned every comment, every mistakes. We have taken a lot of pain to mark out every mistake and write a comment that what was your mistake. So please be careful next time.
Sir,
Yeah.
sir, the last experiment wasn't recorded. So if you could just, record it or just tell us about it again so that everyone can know.
What the last experiment?
So the last experiment class was not recorded.
What was it? You need to tell me.
So I don't remember. I mean,
So I guess the
but they
last time around,
So it's about
Professor just
last
said that.
Monday.
Sir, last to last
Yeah.
Monday. Last Monday was the minor, and the Monday before that.
No,
On
sir.
that day, there was.
So you directly write a mail to Professor Mukherjee, no?
So I guess the last class and the last to last class, like the professor had said that the last to last experiment is long enough that you can actually have it as like two reports. So the last lecture was actually last to last week's report itself, where we had to find the kinetic parameters of enzymes.
Okay, so you write two reports
Yes.
on that, no problem.
Time inhibitors and etc.
That's fine. You make two reports. If somebody wants to make one report, like, yeah, I hope that we haven't, we haven't see, we haven't taken those things seriously. If someone want to file it in two reports, is good. If you want to file it in one report, but obviously what are there, you should cover it up. Many people have submitted only protein purification and that the basic part. They haven't given the enzyme kinetics part in the report earlier. So obviously your marks will not be according to that. So whatever you submit in whole and in full. And you can also consult between your friends that what everybody is doing. No need to you have to write the things on your own, but you can obviously consult because there may be a confusion. I agree, because we are having online sessions and there can be confusions because of many reasons. But you have to be consistent and discuss what are the things that has to be submitted. Got my point?
Yes, sir. Like today, you give 5 questions for us. It would have been better, like in last experiments, you have given these types of questions. Then we have a track like where what we have to do exactly.
Unfortunately, that is what we decide, not you. Okay.
So when could we
Yes,
get the corrected answer sheets and the reports so that we can have a look at them?
I hope I have already talked to Professor Mukherjee and he will communicate with you shortly. I will remind him again, maybe within this week or at the end of this week, maybe.
Sir, when is
And
the
one more announcement. Next Monday there will be no class, so you can have one more week to finish everything, wrap everything up.
sir, when is the minor exam of this course?
Minor exam means you mean there will be a lab test, right? Lab quiz, something like that.
Yes, sir. Sir, professor said that there would be a minor of 20 marks in the starting of the class.
Yeah, so it will be not this week and maybe not next week also, because Monday is a holiday, so there will be, so it will be after that, and he will let you know. I will again remind him that he will let you know, and there will be, I hope, enough time given to you so you can prepare. Okay.
Yes, sir. Thank you, sir.
Okay. So I hope coming back to the today's question, I hope everybody has taken note of the questions.
Yes, sir.
Yes, sir. We have a screenshot.
Okay, so I hope you will all write, be precise and be logical, and also take care of your English and language so that it is understandable to everybody. Okay, so I will end up here. So I hope there is no questions and no things regarding any concept or anything. So I will then quit.
Fine, sir. Thank you, sir.
Okay, so all the best.
Thank you, sir.
Thank you, sir.
Somebody please end the recording.